So what we're going to try to do is finish lecture 1 if we have time into part of lecture 2. Okay. So when we come back, we have 2 weeks and then tests. So it'll be kind of a quick stream toward the evening. So yesterday in lab, we were talking about supporting cells, which are not neurons that function in the capacity of carrying impulses, but support neurons in various ways. So what was unique about the supporting cells in the PNS? They all start with S. So Schwann cells are a critical part of the supporting cells in the PNS, and they line up along axons and create the myelin sheath. And then what we're going to see as we discuss how neurons work today is that the fact that neurons are myelinated and the fact that we have the junctions between the myelin pieces, which are called nodes of Ranvier, that accelerates the rate at which impulses can travel. And so that becomes the key to neurons that are actually quite long cells, is accelerating the rate at which an impulse can travel on the neuron itself. Okay. So we end up with classifying neurons within the PNS 2 ways, either unmyelinated or myelinated. And myelin is created by the wrapping of Schwann cells around the axon. And then we end up with a neural limit to the outside. And then unmyelinated neurons just have a little pocket within the Schwann cell that helps kind of organize the axons themselves. And so again, when you look at a cell, the characteristic that we see is the myelin created by Schwann cells are these halos that we actually see around axons. And then in other axons, we see no halos. So these are unmyelinated axons, myelinated axons. So the second supporting cell was a satellite cell, which are uniquely found in ganglia. So again, the definition of a ganglia is
cell body.
A group of neuron cell bodies where specifically? In the PNS. So again, these are cells unique to the PNS. So the characteristics we looked at in the lab where we looked at the dorsal root ganglia and we looked at sensory neuron cell bodies who are surrounded by these satellite cells. And we can see the images of the satellite cells that surround them. So I have a picture of the same. So as we transitioned into the central nervous system, the brain and spinal cord, then we lose Schwann cells and we lose satellite cells. And so we have a replacement of cells. So we still have myelinated axons as a component of the spinal cord and the brain. And in fact, it's the myelination that creates the white matter appearance of those areas of the central nervous system. So the cells that produce the myelin in the central nervous system are oligodendrocytes. And they again wrap around axons. And they also create the little spaces between myelin, which are nodes of Ranvier. So those are shared characteristics. The other cell we looked at in lab were astrocytes. And astrocytes play a really important role in the three-dimensional framework that creates the brain and spinal cord, and also play a role in the blood-brain barrier. So as the picture indicates here, they can connect to small blood vessels, they can connect to processes of neurons, and they can help transfer material between the two. Plus they create scar tissue in areas where trauma has occurred within the central nervous system. The microglia cells are phagocytic cells that we would find within the central nervous system. So they are responsible for removing bacterial viruses that may penetrate the blood-brain barrier, and also taking care of cellular debris from neurons. So one of the things we know is that in your teenage years, you have the highest number of neurons in your brain, and for the rest of your life, there's a slow decline of neurons in your brain. And so It's the microglia cells that take care of these dead and dying neurons. And then if you do things like consume a goblet of alcohol at one time, so that you almost are comatose, then alcohol accelerates the rate of neuron death, as do certain drugs like meth and other things. So you see an early onset in things like dementia from exposure. And then lastly, the brain itself, when we talk about it, has cavities in it. And what we're going to talk about when we start talking about the brain is how the brain forms embryologically. So the brain actually forms as a tube, and the tube actually has a lumen or a space in it. And as the brain continues to form, we retain the spaces within the brain. And all of those spaces are going to be lined by ependymal cells. And then depending on where we're looking at in the brain for ependymal cells, they have two primary functions. So in modified capillary beds that we're eventually going to talk about called choroid plexes, we're going to see that they actually actively produce fluid that is formed inside of our brain and eventually ends up outside of the brain between the pia mater and the arachnoid mater. So what was that space we looked at yesterday? So the subarachnoid space. And so what we're going to see is that these ependymal cells actually produce cerebrospinal fluid, and then the rest of them have cilia that help move cerebrospinal fluid. So we're actually going to produce this fluid internally within the brain, eventually move it externally to provide a buoyant environment for our brain to protect us from mechanical trauma. And then eventually we're going to return it back to our blood and then constantly recycle it. So it's going to be kind of a fluid process that we're going to look at that involves ependymal cells. All right. So the good news is when we look at neurons, the basic principles are exactly the same as we looked at in skeletal muscle. So the way in which impulses are conducted on a neuron are based upon changes in membrane potential. So what were the three phases of the membrane that we talked about when we did muscles? So if the muscle's doing nothing, we say the membrane's at rest. If we open sodium channels, then we say the membrane undergoes a depolarization. And if we close sodium channels and open potassium channels, then we say the membrane here undergoes repolarization, which ultimately brings us back to resting membrane potential. So the good news is that it's all stuff we've talked about, and we're just going to fine-tune it a little bit for the nervous system and add a little more detail. So hopefully you learned it in muscles so that you don't have to relearn everything for this week. But as we just said, the basic pattern that we talked about in muscles is the same for neurons. So we have a membrane at rest, and a rest membrane would be setting at approximately minus 70 millivolts. And when we talked about muscle physiology, at minus 70 millivolts, there was a charge disparity across the membrane. It was positive on one side, negative on the other side. So at rest, where is the membrane positive, on the outside or inside? It's positive on the outside and negative on the inside. So when we undergo a depolarization, we go from minus 70 to plus 30. And the word depolarization tells us that the membrane is going to reverse its charge disparity, right? So it's going to become negative on the outside and positive on the inside. So the same event is required to initiate this process because we're dealing with voltage-gated channels. And so what we have to do is move the membrane to a key point, which is minus 55. So we say that is a threshold event. And if we reach a threshold event, that's what rapidly opens our sodium channels and causes a rapid depolarization. As soon as we hit plus 30, because we're dealing with voltage-gated channels, then sodium channels are going to close and potassium channels are going to open, and we're quickly going to undergo a repolarization event. So the key to it is understanding that to initiate anything, we have to reach this magic level called a threshold. Once we've reached threshold, then the events are predetermined because they're voltage-gated channels that are going to open and close and control the activity of the membrane. Okay. What's going to become important in neurons that we can kind of gloss over in skeletal muscle was the idea that we actually, during the repolarization event, is actually exceed the resting membrane potential in terms of being more negative than minus 70. And we call that a hyperpolarizing event. And essentially what a hyperpolarizing event does is change your threshold response. So let's say that we're sitting at minus 80 here, but we haven't changed the threshold. So where this is a 15 millivolt change, during the hyperpolarization phase, it would be a 25 millivolt change. So during this period of hyperpolarization, the neuron can actually still be stimulated, but it requires a stronger threshold stimulus to accomplish it. Okay? And then the other thing within neurons that's going to be critically important is that if you look at this period where the light green is and the light yellow, and you look over here, now we also have a period that's said to be an absolute refractory period. So during an absolute refractory period, the neuron cannot be re-stimulated. So it will be non-responsive to any stimulus during this absolute refractory period. So what we then have is kind of two periods in neurons that become important. This absolute refractory period, where the neuron simply will not respond again. And then a relative refractory period, which is during this period that is the dark yellow, that is our period of hyperpolarization primarily, where it'll respond again, but we require a greater stimulus. Okay? So in skeletal muscle, we could crowd stimuli, and we demonstrated it in lab by increasing the frequency of stimuli per unit of time. And we could crowd the stimuli to the point where the muscle would just physically stay contracted as long as you're stimulated. So by having an absolute refractory period, we prevent neurons from being stimulated for long periods of time. So all information flow is pulsing flow within the central nervous system. That's the importance of that. So the other thing we're going to do is we're going to see that we actually have kind of two channel mechanisms that are going to become important as we look at this. Okay. So if we think about the way neurons would work, is we have neurons that have to interact with each other and to other entities. So now I'll just draw the dendrites here. Okay. So what we're going to see is that in this area we're going to have voltage-gated channels. We're going to have voltage-gated channels. But in this synaptic area, we're going to actually have channels that respond to chemicals, so they're called ligand-gated channels. So kind of the simplistic way to think about it is that once we initiate a threshold at this unique place, which is called a trigger zone, then once we reach a threshold within a trigger zone, then we're automatically going to open voltage-gated channels, and the membrane is going to go through a depolarization, repolarization sequence rapidly. When we get to the place where neurons interact, so what do we call the space between the neuromuscular junction and the motor end plate on our skeletal muscle, between the axon ending and the motor end plate? So it's called a synaptic cleft, right? So this space is a synaptic cleft. And how did the neuron communicate with the muscle across the synaptic cleft? They had to release a compound, acetylcholine, which is an example of a broad group of compounds that we could call neurotransmitters. So the same thing is true within your central nervous system. We use voltage-gated channels here to carry impulses along these cell extensions, as long as we create a threshold at the trigger zone. But when we reach this space, we have to communicate across it. And the way we're going to communicate across it is with neurotransmitters. And so the neurotransmitter is going to bind with a receptor on this membrane. That receptor is going to be a ligand-gated channel. Right? So we have voltage-gated channels in particular areas and ligand-gated channels in other areas as we look how neurons interact with each other. So at the trigger zone, if we create a threshold event, then we're going to go from a resting membrane state to a depolarizing membrane state. And sodium channels are going to open. In what direction is sodium going to move relative to the neuron? So we can think of the neuron again as having intracellular fluid. And we can think about the stuff around the neuron as extracellular. So sodium is going to move. Sodium is going to move from where to where? From extracellular fluid to intracellular fluid. Okay. And then as we quickly convert from a depolarization to repolarization, then potassium is going to move. And where is it going to move? From intracellular fluid to extracellular fluid. And what mechanism do we use constantly to make sure that these ions are in the appropriate locations? So we have an antipolar system that we call the sodium-potassium pump, right? So all that's the same. That's the beauty of this is we worked on it all last year. So the good news is if you didn't quite understand it then, you have another opportunity to work on it again and understand it. And then when we get here, we have to use a chemical that's going to move by diffusion. And diffusion always requires a compound to move from an area of high concentration to an area of low concentration. So it's going to move from where it's released to the next neuron in the sequence, and then it's going to bind with these ligand-gated channels. So the cool thing is that's all similar to what we talked about in the past. All right. So if we think about what we just were discussing, then the cell body and the dendrites are blue here because what initiates the response in it are ligand-gated channels. And the cell extension, the axons in this case, are this color because what is involved in impulse travel there are voltage-gated channels. Okay? So we just talked about the fact that we have two neurons. We have neurons where Schwann cells create these blocks of myelin, so that we have myelin and an absence of myelin, which would be a node of Ranvier. And over here we have no myelin, so we would call this a unmyelinated neuron, and we would call this a myelinated neuron. In both instances, we would need to have a threshold effect at the trigger zone, which is going to initiate the carrying of an impulse along the axon. So we have two types of conduction. Continuous conduction, because there is no myelin. and then a skipping conduction pattern, which we call saltatory conduction. Okay. So what do you know about oil and ions? Do oil and ions interact? What do you think? Wow. So you have long weekends, so you can conduct an experiment at home. So just get 2 little glass jars, put water in one, put oil in one, put a couple of tablespoons of salt in both, shake it up, come back on Monday and tell me what happens. The salt in the water will dissolve. The salt in the oil will never dissolve. Because ions work in an environment where there are charges, and water molecules are polar molecules that have charge. Fat molecules are non-polar. So then, if that makes sense to you, then would you assume that sodium and potassium can move through the area of the membrane where the myelin overlaps? The answer is no. Yes. So what actually happens is if we initiate a threshold event at the trigger zone here, then we open sodium channels here. And there aren't any active sodium channels where the myelin is, because this ion won't move through it. So what's going to happen is here's your axon, here's the block of myelin. So what do we know about charged entities? So if I open sodium channels here, a bunch of sodium is going to run in. So does sodium actually like to hang with other sodium atoms? Like charges do what? They repel each other. So even though you're creating all this sodium movement in, they're not going to hang around right here with each other. So as soon as they get inside, what are they going to do? They're going to actually try to get away from each other. So what actually happens is the movement of these ions internally triggers a threshold event at the next node of Ranvier. And as you trigger a threshold effect at the next node of Ranvier, then those sodium channels are going to open. Because they're all voltage-gated channels, the sodium channels at the preceding node of Ranvier are going to eventually close, and potassium channels are going to open. So what actually happens is, at the trigger zone, we create this threshold event because ions are moving, and because electrons move with charged ions, then we actually immediately then begin to set up threshold events at every node of Ranvier in sequential order. So that as one node of Ranvier is undergoing depolarization, the preceding node of Ranvier will start undergoing repolarization and eventually go back to rest. And so this event, where we create something at the trigger zone, which creates a pattern of ionic movement, is going to trigger the sequential opening of sodium channels. So what actually happens is the depolarization event skips from the trigger zone to node of Ranvier, to node of Ranvier, to node of Ranvier. And so by triggering a skipping pattern, we're going to greatly accelerate the rate at which this impulse can travel. And that's the key to myelin, is the speed at which impulses are being carried. So if we don't have myelin, then we still have the same phenomenon going on, that as ions move in, they move away from each other. So we have a way in which we can continue this threshold event leading the impulse, but instead of skipping from node of Ranvier to node of Ranvier, it's going to move continuously down the axon. And so we call that continuous conduction, where you have depolarization and repolarization moving his way down the entire length of the neuron. So the way to think about it is with saltatory conduction, you get to skip. With continuous conduction, you have to walk. So what would be faster? Skipping, yeah. So, and that's the mechanism that we're going to think about. Okay? So putting that into context, then if we go back and look at that one slide where we were looking at cross-sectional views of neurons, then we have what we call type A neurons. And so the type A neurons have nerve fibers with the biggest diameter. And then they have a fairly large layer of myelin that surrounds it. And so type A fibers are the largest diameter myelinated fibers. And they have the fastest saltatory conduction. So 12 to 130 meters per second. So to put that in context, if it wasn't a rainy day and I could find 130 meter sticks, we could go out in 142 meter sticks. We'd go out in the parking lot and we would lay meter sticks end to end, 130, and then we would lay 12. And if we stimulated the type A neuron, it would take a second to travel that distance. But if we stimulated the type of neuron, a different type of neuron, it would take much longer to do this. Pretty amazing. So not surprisingly then, we use these type A fibers for sensory apparatus on our skin and motor responses from the brain. So if over Thanksgiving you were cooking on one of the new glass top stoves, once you turn the burner off, it doesn't glow red, but that glass top stays really hot. So how slow of impulse would you want from the sensory apparatus in your skin to tell your brain that your finger is
burning?
Would you want it really slow or would you want it really fast? You want it really fast because the longer you leave it there, the greater the potential damage to your tissues, right? So you want that instant information that goes to your brain and says, stupid, this hurts. And then what do you want? You want the motor cortex to send a signal back to your muscles to move your fingers. And so you would want that whole pathway, thinking about a reflex arc, from sensory, from a receptor to sensory afferent neuron, to integration center in the brain, to motor efferent neuron, to an effector to be very rapid. Right? So how would you contrast that to your internal environment? Is your external environment much more varied and can have quicker changes in the environment around it compared to your internal environment? So if you just think about the body, there would be areas where you need very, very rapid response times. And then there would be areas where it's much more stable. And so the response times don't have to be as rapid. Yeah. So type B fibers are intermediate in diameter, and they have slower saltatory conduction. So if both of these have saltatory conduction, we're defining them as myelinated, right? Because myelinated and saltatory go hand in hand. And these would be from visceral sensory neurons, so conveying information to your brain about changes in blood pressure, changes in internal body temperature, and things like that, because the internal environment is reasonably stable, and it doesn't have as dramatic change as your external environment. And then type C fibers are unmyelinated continuous conduction fibers. And they're mostly from internal environments as well, with visceral organs. So I just brought this forward for the convenience of lecture. You guys have a few things backwards. So what we're talking about then are these really large ones like the arrow was pointing to, which is a type A fiber. Then intermediate ones like the red arrow is pointing to, which would be a type B fiber. And then fairly small unmyelinated ones right here, which would be a type C fiber. And the commonality for type A and B fibers is that they conduct via saltatory or continuous. Saltatory conduction, which is a more rapid conduction mechanism. And then the type C fibers lack myelin, they're unmyelinated neurons, so they would conduct impulses via continuous conduction. And a basic pattern to think about is your surface can undergo dramatic change. So you can go from a reasonably warm environment here, step outside to a reasonably cool environment where your surface temperature changes quickly, so you want a quick response. But your internal environment is much more stable. So the other way to think about it is to think about the fact that even within neurons, there are differences in the way in which it occurs. So the way to think about it, going back to why this is blue and why this is a different color. So on the graph where I just was, the blue is where we would find a graded potential. And the salmon color here is where we would find a action potential. Okay. So in this area where we have a graded potential where it's blue, we would find no myelin. And in this area where we have this salmon color, we would find myelin if it's a myelinated neuron. So what kind of conduction are we going to have over here where it's blue? Graded, continuous, or solitary? Continuous. And then if we do have a myelinated fiber, then we would have solitary conduction. So we actually have differences in conduction, just depending on where on a neuron we're referring to. So this table just recaps some of the things I just pointed out. So graded potentials are going to occur on dendrites and cell bodies. Action potentials are going to occur on cell extensions, axons, and they're initiated at the trigger zone that we talked about. What initiates a graded potential are the receptors at the dendrites that are being stimulated by a neuron that was preceding it, and they respond to neurotransmitters. So those receptors are called ligand-gated channels that are going to initiate a graded potential. And because it is initiated at a trigger zone by a threshold effect, then what we would get with an action potential traveling on an axon, what we would get would be an action potential. So graded on the axon, graded on the dendrite cell body, and then an action potential on the axon. So let's just review a couple of other things that we talked about. So duration. What we said with type A and B type fibers is they have very fast transmission, and each node of Ranvier stays depolarized for a very, very short period of time. So the duration is very short. 5 to 2 milliseconds. Whereas if you don't have nodes around the air, then it's a much longer conduction period. You have a longer time of duration. All right. So now let's think about propagated and the word propagated. If you propagate something, if you're a botanist, you would make a new plant off an old plant. So you're maintaining something, right? So the word propagated in this instance means that at the trigger zone, if you open sodium channels, you're always going to go from minus 55, which was the threshold, to plus 30. And it doesn't matter whether you're at, and we have our meter sticks out in the parking lot. We have 130 meter sticks end to end. Doesn't matter whether you're at meter stick 60 and 61, 90 and 91, or at the end at 130. Every event would be a shift from minus 55, which was our threshold, to plus 30. That's what propagated means. So we have propagated saltatory conduction and action potentials, which tells us that even at the end of 130, we're still going to go from minus 55 to plus 30. So if it's non-propagated, what it's telling you is that it fades over distance. So we could actually start at the dendrite, where we had enough neurotransmitter released and bind with enough receptors so that we go from minus 55 to plus 30. But by the time we get partway through the cell body, we could be going from minus 55 to plus 1. Okay. So the significance of non-propagation is that with distance traveled, the impulse can fade. So graded impulses can fade. And then the importance of that would be if we're looking at that event, and here's our dendrites going into a cell body, and here's this important point on the trigger zone. Then we can start here with a shift from minus 55 to plus 30. But because it's not propagated, by the time it gets here, it could be minus 55 to plus 5. And then as it travels further, it could be minus 55 to minus 25. And then as it travels here, it could be minus 50, it could be minus 60. So if we don't reach minus 55 with the trigger zone, we will never trigger an action potential. So a graded potential over here becomes important because as they fade, it's possible to reach the trigger zone at a lower than threshold level. And if you reach the trigger zone at minus 60, you will get no action potential. So then we would have to release more neurotransmitters over here to get a stronger response. So it's a beautiful way in which neurons can interact with each other and get responses and non-responses. So there's a couple of ways to think about this. It's easy for the way the nervous system to work to think we always want positive outcomes. But we have a lot of areas in our brain that work by inhibition, where we actually inhibit outcomes. And if we inhibit outcomes, then we have normal events going on. Where if we don't inhibit the outcomes, we have abnormal activity that begins to occur.
Just going to say it sounds like someone dares you to do something and then you continue to stand there thinking, okay, am I going to do this? Am I going to do this? Or am I going to do this? And then not until you...
Yeah, you're standing on on a skyscraper downtown. Somebody dares you to jump on
If you first thought, if you acted on your first thought, you would have jumped.
it. Yeah, very interesting, indeed. Okay, so what I did is I actually put that idea of conduction, whether it's propagated or not propagated, together with amplitude. And so over here, if we reach threshold, we're always minus 55 to plus 30. And it doesn't matter how far we travel, it's always going to be minus 55 to plus 30. So that is a propagated event, and the amplitude is an all or nothing event, meaning that you're either going to get a minus 55 to plus 30, or if you don't reach threshold at minus 55, you get nothing. Does that make sense? So it's all or nothing. If you reach threshold, you're going to go to plus 30. If you don't reach threshold, you're not going to do anything. Whereas a graded potential is not propagated, therefore its amplitude can change. Does that make sense? That's what we were saying here, is the amplitude is changing with distance traveled. All right. And then the interesting thing when you look at the pattern is, remember during a refractory period, we either can't get another stimulus, or we can get another stimulus, we can get another event, but the stimulus has to be greater. Okay? So in an absolute refractory period, the neuron itself cannot respond again, which ensures that you can't do wave summation like we did in skeletal muscle. And then where this is going to be interesting when we talk about it is in cardiac muscle. Would you want wave summation in cardiac muscle? Would you want your heart to undergo complete tetany for any period of time? No, because it would just stay contracted. And if it's contracted, it ain't pumping. It's pumped once, and it's not going to pump again. So what we're going to do is begin to tie this eventually together to some real-life experiences. So here, there is no refractory period. So you can actually have summation. So what's pretty amazing when you look at the two events is that you can summate here and assure that you get a minus 55 at the trigger zone. But over here, you can't summate, and so you always have pulsing impulses that actually occur. And we're going to look at a pattern to that here in a minute. So we're building a basis that we're going to look at some things with. Okay, so if we look at how neurons interact, then the way in which the place where neurons interact is a synapsis. So that's the synaptic cleft, all right? So if neurons are actually in physical contact with each other, and they have communication junctions called gap junctions, then you don't need a neurotransmitter. If there's a physical space between the two neurons, then you need some way to convey information across the space. So some areas in our brain we use electrical synapses, which is communication via gap junctions. And because you have two neurons actually interacting with each other, you can have two-way flow of communication. In other words, neuron A can communicate with neuron B, or B can communicate with neuron A. So it provides very fast, synchronized, two-way transmission of information. And it's critically important to cardiac muscle. It's important to smooth muscle. And it's important to neurons in the central nervous system, particularly where memory is stored. Okay? Because you have to create a memory trace, and then you have to recover the memory. All right? Chemical synapses, because we're releasing a compound that moves by diffusion, and diffusion is always from an area of high concentration to low concentration, creates one-way flow of communication. So we use chemical synapses, or we use neurotransmitters that move by diffusion as an effective mechanism, but it creates one-way flow of information. So the information is always going to flow from neuron A to neuron B, and not vice versa. So the way we control skeletal muscle is we have neurons that arise on our cerebral cortex, on the frontal lobe of our brain, and their axons go down to some level at your spinal cord, depending on whether you're talking about muscles of the arms or muscles of the legs. And then a new neuron in the gray matter in the spinal cord then has an axon that goes out and becomes part of the peripheral nerve. So every skeletal muscle contraction involves at a minimum 2 neurons. And these neurons communicate via neurotransmitters. So it's always one-way flow. So it goes from the cerebral cortex out to the skeletal muscle. But it doesn't go from the skeletal muscle to the cerebral cortex. So it's one way. So if we think about it, what we've just been talking about, then we create an axon potential that travels down our axon. And so we'll call this neuron A, or we could call this the presynaptic neuron, because what we're doing is looking at a synapsis, which is the way two neurons interact. And so if this is a dendrite, then would this be a gradient potential or an action potential? Great. Now, you can correct me. Oops. So we would have an action potential here, but we would initiate a gradient potential there. All right. So the first thing we want to do is add a new little piece of information, which is how we cause the release of neurotransmitters. So how does this event, we call an action potential, which is depolarization, repolarization, actually cause neurotransmitters to actually be released? Okay. And what happens is we have unique channels in our synaptic involve, which are calcium channels. So as this wave of depolarization reaches the synaptic involve, instead of continuing to open sodium channels, we're now going to open a new channel called a calcium channel. Okay? And then calcium is going to be more concentrated in our extracellular fluid. So we're going to have an increase in calcium out here compared to our intracellular fluid in the synaptic involve. So if that's true, then calcium is going to diffuse in. All right. So if you're in a clinical environment, we have two words that we would describe for somebody's blood calcium levels. And blood calcium determines your level in your extracellular fluid. Right? So if you don't have enough calcium, you would be in a state of hypocalcemia. And if you have too much calcium, you'd be in a state of hypercalcemia. Now, drawing from Unit 2, where we talked about bone physiology, where we normally store calcium is in our bone matrix. So normally, if we would enter a state of hypercalcemia because we ate our sardine yogurt for breakfast, then the thyroid gland would release what hormone? Calcitonin, which would cause osteoblasts to take the extra calcium deposited in our bone. If we decided we didn't like our sardines with yogurt, and we didn't eat any calcium at all, if we decided we were going to fast for a while, then our blood levels of calcium would eventually drop. And so what endocrine organ would be involved? The one that sits on the back of the thyroid gland, parathyroid gland. and the parathyroid gland would release PTH, and that would cause osteoclasts and to some extent osteocytes to release calcium from the blood. So we have a mechanism where we try to maintain blood calcium at a level we would call homeostasis. So if your blood calcium is within the normal range, you're in homeostasis. If it drops below, you would be in a state of hypocalcemia. If it goes above, you'd be in a state of hypercalcemia. In a state of hypocalcemia, if you were doing reflexes, tests on somebody clinically, they would have slow reflexes. If you were in a state of hypercalcemia, they would have hypersensitive reflexes. And then there would be a bunch of neurologic stuff going on with them as well. It's because we require calcium to make neurons communicate. Now, the mechanism that is a little harder and we don't quite have the complete picture on is how moving calcium into the synaptic involve causes excretory vesicles with pre-made neurotransmitters to actually migrate to the synapses and release the neurotransmitter. But we know a buildup of calcium does that. Now we know if you can't build up enough calcium, then you don't release as much neurotransmitter. So we've learned with certain things like manic depressive disorders, we can use ions like lithium to actually influence the way the nervous system's actually working. So we can use other ions to play a role as well. Okay. So the whole point is that we're going to release neurotransmitters. They're going to bind over here. So what kind of channel is this? It's a ligand-gated channel. The thing we're going to talk about is we got a whole suite of neurotransmitters. And for it to work, the ligand-gated channel has to be unique for a particular neurotransmitter. So if we have a ligand-gated channel that normally binds acetylcholine, it will not bind epinephrine, norepinephrine, and the other neurotransmitters we've been talking about. So there's a unique connection between the ligand-gated channel and the neurotransmitter that it responds to. So they're unique. So if this is acetylcholine, this would be acetylcholine ligand-gated channel. And if we bind enough acetylcholine with these channels, and we open some sodium channels, then we're going to get a graded potential. If that graded potential reaches the trigger zone at a threshold level, then the graded potential would be converted to a action potential. Right. So I want to go back to another statement here that I just kind of avoided for a little bit. That was that at the trigger zone, if we have a threshold, we're always going to get a depolarization followed by a repolarization that takes us back to rest. Well, what we can get at a dendrite with a graded potential is a depolarization or a hyperpolarization. So if we hyperpolarize the neuron at the dendrite, then we're not going to move it from minus 70 toward minus 55. What are we going to do? We're going to move it from minus 70 toward minus 80 or some other event. So we actually have neurotransmitters that can create a positive event, and we have neurotransmitters that are inhibitory. And then that facilitates a large way in which our brain can respond to certain things. All right. So, for example, if we have sodium channels, and if these are ligand-gated sodium channels, and the neurotransmitter binds with it, we increase our sodium content, then we would create what we call an excitatory postsynaptic potential, or an EPSP. And so we would want that area of the brain to actually become active. We have different channels that these compounds can bind with. So we could actually have calcium channels. And if the neurotransmitter binds with it, then we move calcium from our extracellular component to our intracellular component. But not calcium, excuse me, chloride. But chloride is a negative ion. So if I'm adding a negative ion to the intracellular fluid, I'm going to make it more negative on the inside. So I'll move the membrane from minus 70 to minus 80 to minus 90. Now we create what I call an inhibitory postsynaptic potential. So the way the brain works is you want to shut some areas of the brain down, and you want other areas of the brain to be activated at any point in time. And so you have to have a way in which you can create these inhibitory events, and you can create excitatory events. So let me give you a great clinical example of it. Most of us have no problem holding our hand up and keeping it steady. Somebody has Parkinson's disease. What do? They tremor. And then what happens when they want to do something like pick a spoon out is the tremors actually get even more violent when they're trying to facilitate muscle contraction. So there's actually an area of our brain that controls that. And in normal physiology, the area of the brain is constantly being created, we're constantly creating inhibitory postsynaptic potentials that make those tremors not occur. So if that area of the brain stops working, and you stop the inhibition process, then you get these spontaneous muscle contractions or tremors.
So they fix that by introducing a lot of negative ions?
Pardon?
Would they try to help that by introducing a lot of negative ions? Or?
Well, no. What we do, we actually, there's two ways, which is really kind of cool. So the way we've done it for years, once we began to understand this process, is we learned that there was a critical neurotransmitter. So the neurotransmitter is called dopamine. And you got to have adequate dopamine levels to inhibit the neurons in an area of the brain that we're going to learn next week called the basal ganglia. Okay. So what we know is that for some reason, another critical area of the brain called the substantia nigra stops making dopamine. And as dopamine levels drop, then the tremors occur. So what we've used historically to control them is dopamine. We give the patients dopamine. And we give it in a long-acting form and a short-acting form. So carbidopa, levodopa, two different forms of dopamine. The dilemma we have is we have an enzyme in our blood that breaks down dopamine. So we got to give the patient crazy amounts of dopamine to get some into the brain to actually control Parkinson's. So when somebody is initially in Parkinson's, the dopamine seems to work pretty well, and they can moderate their tremors some. But as Parkinson's continues to manifest, then it's harder and harder to use L-dopa and carbidopa to control the tremors, so the tremors get worse and worse and worse. And then eventually you get spasms of swallowing muscles and stuff that leads to death with Parkinson's. So it's a terminal disease. So what we've learned, which is really cool, and I saw it in the early 90s at Toronto School of Medicine when I was there at a meeting where they had a patient who was sitting there just shaking like this. And what they had done is they had used long needles to stick little electrodes into the area of the brain. And they had this little box under the skin here that they could control with a magnet, toggle switch on and off. And so at first it's off, and the person's just, and then they turn it on and the person goes, and it completely stops. And so we've learned that we can use weak electric stimuli to that area of the brain to maintain those inhibitory impulses. They just started doing that surgery here in Spokane about 5 years ago, 6 years ago, where we can use electrical stimulus to control Parkinson's. Yeah. So anyway, so the take-home message is, in your brain, everything doesn't have to be positive. In fact, it's critically important that some things are actually negative. And so we want to create EPSPs in certain areas, IPSPs, in other areas to create what we can consider normal functionality. So just to kind of reiterate, so in an EPSP, we want an event that moves us toward threshold so that we can get an action to control. To create an IPSP, we actually want to go the opposite direction, which would be a hyperpolarizing event that would take us further away from threshold. And then we can inhibit the neuron itself. So what I want to do is I want to use some examples of neurotransmitters, and I want to show you how neurotransmitters play in this arena of creating IPSPs or EPSPs. So the thing you have to understand, and we're going to fine-tune this as we go forward eventually, is that we can have more than one receptor for a particular neurotransmitter. So that in some areas of the body, a neurotransmitter creates an IPSP, but in other areas of the body, the neurotransmitter can create a EPSP. And so we have different types of receptors. Now, we've done a tremendous amount of work with this for receptors that respond to epinephrine and norepinephrine because it helped us control blood pressure. So we know we have alpha receptors, and we know we have beta receptors for norepinephrine. And we know if we can influence beta receptors by using beta blockers, we can control blood pressure. So some of our earliest, strongest blood pressure medicines were called beta blockers because they would block the ability of beta receptors to bind with acetylcholine to create increased cardiac output, which created heart attacks over time. Okay. So we could control blood pressure. So there are two events in medicine that have led to three events in medicine that have led to the increase in longevity we see today compared to the early 1900s. The ability to control blood pressure, key to elongating life and avoiding early strokes. The ability to control blood cholesterol to prevent heart attacks, and the advent of antibiotics to keep us from dying from bacterial infections. Those three events have elongated life significantly in developed areas of the country where it's readily available. So acetylcholine, which is found throughout the body in the PNS and CNS, can actually create EPSPs in some areas and IPSPs in other areas. So that just tells us we must have more than one type of receptor. Okay? There's kind of a cool one that we use clinically. And so there are some amino acids that act like neurotransmitters. So glutamate and aspartate are two examples of it. And then there's a modified amino acid called gamma-aminobutyric acid, or GABA. And GABA is critical to producing IPSPs in the central nervous system. So, and we use it to control anxiety. So if you're a very anxious person all the time, it means that part of your brain that normally should be creating IPSPs isn't. And therefore your brain is just always hyperactive. And so we created a compound called Valium that actually enhances the effect of gamma-aminobutyric acid. So if you give somebody Valium, they become much more calm. So we use it before surgeries when people are really anxious. Because when your blood pressure is way up, you're not a good candidate for surgery. So if we can make you less anxious, keep your blood pressure lower, we can get through the surgery better. So a lot of times we give people Valium before surgery just to calm them down. In fact, when they do LASIK surgery on your eyes, the only thing they give you is Valium to calm you down. So, pretty amazing. If you've ever seen somebody on Valium and you know them, the difference in their behavior patterns is pretty amazing. And so that's pretty cool. So we have some compounds called biogenic amines, and the ones we just talked about, norepinephrine and epinephrine, typically create EPSPs in the sympathetic nervous system, but they create IPSPs in the other branch of the autonomic nervous system. which is the parasympathetic nervous system. So we're going to put that all together for you at the last unit, last lecture of the quarter, and talk about these ideas of how we do that. And then the other one is serotonin. And what we know is serotonin is produced by a little gland that we are going to talk about in the brain called the pineal gland. And what we know in animals is that serotonin levels vary seasonally, and then the serotonin drives reproductive activity. So what we know in elk and deer this time of year is they're rutting, and they're trying to make babies, and their serotonin levels are really high. So there was a recent study done looking at serotonin levels in humans, and they used 3 categories of individuals in the study. Couples that identified themselves as being deeply in love, couples who identified themselves by getting divorced, and then individuals who weren't in a relationship. And what they found was couples that were identified themselves as being deeply in love had the highest serotonin levels. So serotonin not only makes elk and deer want to procreate, it probably has an importance on our behavior patterns as well. So, and then what we do, and we're going to talk about in more detail, is serotonin builds up during the day. We have an enzyme that breaks serotonin down and converts it to another compound called melatonin. But the enzyme is light limited. So during the day, when light is entering your eyes, the enzyme is inactive. And as we go into dark periods, the enzyme becomes activated and serotonin levels drop. So serotonin is what makes you wake up and keeps you active during the day. The drop in serotonin and elevation in melatonin makes you want to go to sleep. Yeah, so it's directly involved in sleep induction. So now we know if somebody has insomnia, which is where they can't get to sleep well, we can give them melatonin and they can actually go to sleep. Okay. So we have gases that are released by neurons as well. So the most obvious one is nitric oxide. And so this is kind of a cool story. I believe it was Eli Lilly, had spent an enormous amount of money looking at blood pressure medicines. And they worked on this blood pressure medicine that seemed to work really well in animal populations. And when they got to the point where they figured out it was safe enough to try on human studies, they got some people with high blood pressure to undergo these clinical trials with it. But all the men got erections. So it was like, this might not work well for their blood pressure medicine. So when you spend a lot of money creating a drug, and the end result is you don't think you can use it, if you find another use for it, and it's still marketable, then you recover your money from your research. So now we sell it as Viagra for erectile dysfunction. Yeah, and it was an excellent drug in decreasing blood pressure because it caused vasodilation, which decreased peripheral resistance and dropped blood pressure. So we now use it like in high elevation events where people get cerebral edema and pulmonary edema. We can use it up there, and it actually brings their blood pressure way down and relieves the edema. But they're so physiologically stressed that you don't get really. Then there's this cool stuff that's called substance P. So P stands for a group of compounds that are prostaglandins. We've grouped about, I think, 28 or 29 compounds into prostaglandins. And we know that some prostaglandins are smooth muscle contractants. And we know that other prostaglandins enhance pain. So if the prostaglandin levels are high, the cognitive capacity of the brain to tell you you're in pain is higher. And so what we have worked on, there are drugs that will prohibit the production of substance P so we can decrease the pain reception So the earliest drug was aspirin as a drug that decreased prostaglandin production and decreased the enhancement of pain. We've come up with some others. Our body actually produces a normal one called endorphins. And endorphins tend to inhibit pain. So that's the body's natural mechanism to do it. And then I was looking at science a couple of years ago, and I was looking at taste buds, because we talked about taste buds in the next couple of weeks. And they were looking at how we control taste sensations. And they did a clever experiment where they altered the production of ATP, which we thought typically is only a compound used to actually provide energy for us. And what they were able to show is if you limited ATP, you converted taste buds to not being able to send any signals for taste. So it kind of led them to believe that ATP is kind of the neurotransmitter between taste buds and the sensory neurons that carry the sensations to pain. So it's kind of an interesting study done in 2005. I haven't followed up on it. I need to follow up on it. So to kind of review what we've been talking about, if we get a threshold event at a trigger zone, then we get a action potential. When the action potential reaches a synaptic end bulb, instead of continuing to open sodium channels, we'll begin to open calcium channels. And what we know is that influx of calcium is responsible for the release of neurotransmitters. Now, what I didn't do as we went through the neurotransmitters was kind of lead you through neurotransmitters that are charged versus neurotransmitters that are non-charged. So, because membranes are bipolar molecules with a charged entity toward the outside and inside and non-charged toward the middle, the membranes have a charge on the outside and inside, but they're non-charged through the middle. And that determines what can actually move through a membrane. So the larger the molecule, the more non-polar charged it has to be moved through a membrane effectively. Right? So what we have then is two mechanisms by which we can actually move the things through the membranes. So we can have a release of a neurotransmitter that binds with a receptor that's an ion channel. And we have different mechanisms by which we could do it. So we could open sodium channels, which would lead to move us toward threshold. We could actually open potassium channels, which would move us away from threshold. So potassium would be another way we could hyperpolarize. Because if you're moving positive out, the inside is becoming more negative. Right? So this is a classic mechanism where we actually have a ligand-gated channel that's an ion receptor, and we create this again. Some neurotransmitters actually bind with a receptor, and the receptor creates an internal messenger. And so this internal messenger is a G protein. And then the G protein activates activity within the cell. So the way to think about it is the neurotransmitter is a primary messenger. And if the primary messenger activates the sodium channels, then that's all you need. You only need one messenger, which is a primary messenger, to elicit an activity in the cell. If the neurotransmitter, and the ones that behave this way are ones that play like hormones and neurotransmitters, binds with a receptor that's not an ion channel, then you have to create a secondary messenger inside the cell to make the cell do something. Okay? And those are two broad mechanisms that we'll look at eventually. But we're going to spend most of our time this unit with this classic of ion channels. All right. So the other thing that is pretty fascinating is to look at the way neurons behave in our central nervous system. So the way our brain and spinal cord work is we have neurons that interact with each other. So we can have patterns to the way in which neurons interact with each other. So the top two are opposite pathways. So the one is where you start with one neuron at the input point. So it's the one that's going to initiate the initial trigger response and get an action potential. And then it interacts with increasing number of neurons. So that we end up with many neurons at the output. So it's called a divergent pathway because you're going from one neuron to many neurons. This is actually the way memory works in the brain, is you create sequences of pathways between neurons that allow us to somehow retain memory. We still don't have memories retrained. We understand that pathways exist, right? So if I want to recall the memory, I have to reverse the pathway. And so that would be a convergent pathway, where we start with many and we end up with one. So two opposite pathways. Reverberating pathway is a real interesting one. So what we do is we have a sequential way in which neurons interact, and then this one interacts with this neuron, and it continues it pathway. So the way you could think about this one would be, if you've ever dealt with patients with Alzheimer's or dementia, they will get into repetitive behavior patterns that just repeat themselves every 3 or 4 minutes. So as a care provider, it totally drives you crazy because they're in this, and you're trying to get them to go somewhere else, but they don't have it. They just constantly do that. So kind of an interesting pattern. We use this with computers. So with a computer, you can set up data sets. You can have the computer actually generate data by looking at different data sets. And you tell the computer to go back to data sets and do it again. So I remember in its infancy, back in the dark ages when I was in college, I took a programming class. And so what we had to do is we had to write these programs. So one of the programs we had to write was a data set program where you had the computer look at different data sets simultaneously. And so it was kind of a reverberating pathway. So I was kind of annoyed toward the end of the quarter and started the class. So I created my data sets. Were all called Hell. And then the password that told me to go to my data set was go to hell. Which I thought was kind of clever, but my instructor didn't think it was. Go to hell 1, go to hell 2, go to hell 3.
How many levels did your hell have?
I think there were 6 data sets.